Hello everybody, the topic of my presentation is High Performance Liquid Chromatography HPLC. I have structured my presentation as follows. First I will define the HPLC, then I will show you the HPLC system, talk about the separation goals, describe the components of the HPLC and the elution types, and finally talk about data calculation. So HPLC stands for High Performance Liquid Chromatography, but it could equally stand for High Pressure Liquid Chromatography. High performance refers to high speed, high resolution separation, and this high performance is achieved by using very small diameter particles of less than 20 micrometer, but usually between 2 and 5 micrometer column packing material to allow large surface area for more interaction and high resolution. So the use of small diameter packing offers large surface area of the stationary phase, which means more interaction of the components with the stationary phase and thus better resolution, high resolution. However, when small particle packings are used, high pressure are required to push the mobile phase through the column. For this reason, HPLC is sometimes referred to as high pressure liquid chromatography. So here you see an animation about separation and detection in HPLC. Two components will move with the mobile phase inside the column over the stationary phase and will make interaction with the stationary phase to a different extent. So they will separate from each other and travel with different speed and will come out of the column one after another and reach the detector and be detected, give a signal which we call a peak. And here about the separation goals. There are three goals for the separation: either quantitative analysis, which is the most frequent one in chromatography, in which we correlate the peak area or the area under the curve to the concentration of the substance, or qualitative analysis, in which we identify unknown substance by matching it to the retention time of a reference substance. Both 1 and 2 are referred to as analytical chromatography, and goal number 3 is preparative separation, which is usually done on larger scale using larger columns to separate components for further use or to purify a substance. The essential components of a complete HPLC system are listed here: solvent reservoir, solvent delivery system, pump, fixed volume injector lobe or autosampler, packed column, detector, and data system and record. And here you see a simple schematic diagram of a basic HPLC system. So number 1 is the solvent reservoir or solvent container. Number 2 is the solvent delivery system, the pump. Number 3 and 4 are the sample introduction unit, either autosampler or manual injection. Number 5 is the column, the heart of the HPLC. Number 6 is the detector, and number 7 is the data system. So first about the solvent reservoir or solvent container. Different containers are used to deliver the solvent. The best material from which the containers are made is glass. Plastic containers are not recommended as it leads to plasticizer leaching. The container should be covered to prevent solvent evaporation. One can have up to 4 containers because the mobile phase can be a mobile phase system consisting of a binary mobile phase, so 2 components, 2 solvents, or tertiary or quaternary, so 4 components, 4 solvents mixed together. The tubing from the container can be made of stainless steel or Teflon, and both are satisfactory. And now about the pump, the solvent delivery system, the component which is shown here below in the system. We usually use constant volume pump, which is mechanically driven system to deliver constant volume. And now about the sample introduction system, which is used to deliver the components of the sample by the stream of the mobile phase to the stationary phase inside the column. One can use either manual injection or autosampler. Autosampler is the part shown here in the instrument. Usually we have a tray in which we can place different vials, small vials, which are used to deliver the component of the sample. So we put the components of the sample dissolved in this vial, and there will be an automatic needle which will deliver certain volume and inject the volume in the stream of the mobile phase going to the column to the stationary phase. Important aspects in sample introduction are precise and reproducible injection. Precise means that you deliver exact amount. So if we would like to inject 10 microliter, the system should inject exactly 10 microliter. And reproducible means that if we repeat the injection several times, the system should exactly inject the same amount every time. And now about the column. The column is usually referred to as the heart of the HPLC system. In fact, it's the most important part of the HPLC system, and it's the part in which the real chromatographic process is taking place. So the components will separate inside the column, will come carried out by the mobile phase and will make interaction with the stationary phase inside the column, and will separate and then leave the column individually to the detector. The column is usually placed in a column oven. This is the part which is shown here, and we have different types of the column depending on the chemistry of the stationary phase, but also depending on the specification, the particle size, the length of the column, the internal diameter, and even depending on the external material from which the column is made of. So sometimes of stainless steel and sometimes of a Teflon-like material. There are different HPLC column types according to the chemistry of the stationary phase, including but are not limited to normal phase silica columns, reverse phase silica columns, ion exchange columns, size exclusion columns, affinity columns, and chiral columns. Now about the separation mechanisms, we have different mechanisms. So we have reverse phase chromatography, in which we have reverse phase silica for non-polar compounds, and normal phase chromatography, so normal phase silica for polar compounds, chiral chromatography, so chiral stationary phase for enantiomeric separation, affinity chromatography, so affinity phase for antigen-antibody interaction, size exclusion chromatography, so dextran-like material for macromolecules like polypeptides and proteins, and ion exchange chromatography, charged stationary phase for ionic compounds. Now about column specification. In fact, there are three important specifications for the column, and these are the particle size, which is usually between 2 and 5 micrometer, the column length, which is usually between 25 and 250 millimeter, and internal diameter, which is usually between 2 and 4.6 millimeter. Another two important issues to consider about the column are, first, the column should never be dry, because when the column dries, the particles of the stationary phase will shrink, and this will result in a void volume inside the column, and the void volume will result in band broadening, and band broadening will negatively affect the resolution between two adjacent peaks. The second point is that columns should be equilibrated before use, and this is usually done by passing about 10 to 20 column volumes of the mobile phase through the column. Now about the detector. In fact, in HPLC, one can use several detectors to detect the separated compounds which elute out of the column. One can use, for example, ultraviolet visible detector or the related diode array detector, fluorescence detector, electrochemical detector, refractive index detector, or mass spectrometer detector. In fact, UV visible detector or the related diode array DAD detector are the most commonly used detectors in HPLC, because most organic compounds show some useful absorption in the UV region. And now about data handling and analysis. In fact, data handling and analysis is currently easy because of the presence of advanced computer softwares. The output of HPLC is a chromatogram, as you can see in this figure. And the chromatogram is a plot of peak intensity against migration time in minute. And from this chromatogram and the peak parameters, we can handle and calculate the data. So here you see the peak parameters. Important are the retention time, which is the time taken by the analyte to reach the detector after sample introduction, the peak area or area under the curve, which is usually directly proportional to the concentration of the analyte in a certain linear range, and the width of the peak. The retention time of a substance can be used for its identification by comparing the retention time of unknown substance to the retention time of a reference material under the same chromatographic conditions. The concentration of unknown substance in HPLC can be determined by correlating several concentrations of a reference substance to their peak area and creating what is called the calibration curve, and then by matching the peak area of the unknown substance to a certain concentration through this calibration curve, the concentration of the unknown substance can be determined. There are three elution types in HPLC. The first is called isocratic elution, in which the composition of the mobile phase remains constant and the flow rate remains constant. The second is called the gradient elution, in which the composition of the mobile phase changes during the run, and the elution power of the mobile phase changes also during the run, usually increase, to elute late eluting peaks. The third one, which is relatively new, is called flow programming and is only possible with columns that allows high back pressure like the monolithic columns. So in this type, flow rate of the mobile phase changes usually increase during the run according to a defined program, to allow fast separation in a few minutes. An important parameter to consider in HPLC is the number of theoretical plates, also called the column plate number or the plate number, and is a dimensionless number that measures the column efficiency and can be calculated according to the following equation, considering the retention time and the width of a certain peak. And in fact, in general, the increase in the n value means higher column efficiency, and the longer the column, usually the larger or the higher the n value or the number of theoretical plates, and so means the higher the efficiency of the column. Another important parameter to consider in HPLC is called the resolution value, RS value, and it determines the degree of separation between two adjacent peaks and is usually calculated according to the following equations, considering the retention time and width between two adjacent peaks. A minimum value of 1.5 is required for full resolution between two peaks, but a value of 2 or more is preferred for robust method. Another important factor to consider in HPLC is what we call the asymmetry factor. In fact, symmetric peak enhances good resolution. Peak asymmetry can be calculated according to the following equation, considering the two half peak widths at 10% of the peak height. If value of more than 1 means peak tailing and of less than 1 means peak fronting. Thank you for your attention.